You all have a good weekend? It's nice weather. Wasn't snowing. Relatively nice weather. Wind's picking up. How's everybody feeling about Wednesday? Terrible? Not good? Okay. Well, we got to change that. So exam 3 is Wednesday. There's a review session this evening, 6 o'clock. Did I get that right? By Zoom. Okay. We'll probably have a little time at the end of today's lecture, or for the remaining time of today's lecture, we'll have some time to do some review. Talk a little bit more specifically about the exam, okay? Why did the man freeze his money? He wanted cold, hard cash. Well done. Why do frogs make good outfielders? Why do frogs make good outfielders? They're great at catching flies. What did the baby corn ask the mama corn? Where's popcorn? Nice. All right. So this afternoon, we're going to finish out central nervous system, and then we're going to switch into review mode. And we'll talk a little bit about the exam. And my hope is that it's more audience participation than Dr. Keller mediated. Well, let's get through some of this material. This is where we left off last Wednesday on the lumbar spine. We talked about the cervical, the thoracic, and the lumbar. And today's material is fair game for the exam. So we want to finish that out, and then after the exam, we're going to get into peripheral nervous system, and we're going to get into muscle physiology, which is really cool. Okay, but again, having a foundation in nerve physiology is really important for understanding muscle physiology and how the muscle contracts. So if we get down here to the lumbar spine, we're in the lower back, and the load stress that's placed on the lumbar spine is the greatest of all the areas of the head, arms, and trunk, because it's taking the whole weight of the head, the shoulders, the muscles that are attached there. You're going to be in lab this week on head, arms, and trunk, and you're going to be looking at those muscle groups. Well, all of them attach either superiorly or to the spine or inferiorly to the lumbar spine, and it's taking all of that weight of the head, arms, and trunk. So when you think about the muscle groups in lab that you're going to be looking at this week, all of the weight of those muscles are bearing down on the lumbar spine. And our regular daily life, your future patient or your current patient, some of you have shared with me that you actually treat patients now. You're CNAs and you're watching patients on a regular basis. And our older patients that are maybe immobile, maybe slightly overweight, they're a little bit more frail. These are areas, and these common activities of just bending over and picking stuff up is really complicated. And if you're a CNA and or occupational therapy, this is not the way for your older patient to pick stuff up, right? That's a super compromised position. So what happens here? Let's talk a little bit about what happens in the lumbar spine this afternoon. We need mobility here because we've got to have the ability to do daily activity, but we also need strength. And so in lab, you looked at the vertebral column, and you look at how thick the lumbar vertebrae are. And as you move from the cervical neck region to the thorax to the lumbar region, the body of the lumbar vertebrae is much more pronounced, much thicker, because the load forces that it sustains is much greater. But we have this balance between strength and stability and mobility, because you need to be able to do daily activity. So when you consider the magnitude and complexity of these functional demands that happen on a daily basis, it's not really a surprise that we get a lot of injuries here. So we'll talk a little bit about these, and the first one I'm going to talk about, these are kind of the two major ones that we'll test on, okay? There are others, but these are the ones that you're going to come in contact with a lot of your patients. Herniated discs. Some of you have had herniated discs, or dads had a herniated disc, moms had a herniated disc, or grandparents. So what are these herniated discs? Well, between these vertebrae, we've got these discs that act as shock absorbers or cushions. And if you move down into the sacrum, which is a portion of the tailbone, if you will, sacrum and the coccyx, those discs are gone. They're gone during embryonic development, and these bones fuse. And as patients get older, this inset is blown out over here kind of in the middle of the screen. As patients get older, the disc health deteriorates. And it's not uncommon for the intervertebral space to narrow, and you lose the disc altogether, and you get fusion on bone. In some cases, they'll come in and they'll fuse the vertebrae all together, and they'll do a vertebral fusion. And that's not what we're really talking about. We're talking about herniated discs, where if there's a lot of pressure here, and it's a bending, twisting motion, it's not uncommon for trauma or damage to happen where you get a bulging or a slipped disc. All these are kind of different terminologies for really the same disease. And it impinges the nerve root. And this is where the pain sits. Or that's what the mechanism, the pain response, is putting pressure on that nerve. So, surgery. Now, you can unload the disc. You go to physical therapy in younger patients, active patients. This isn't a bad treatment regime where you can unload the disc, you can strengthen the musculature, the musculoskeletal system around it. You can get inflammation to drop. Maybe there are some corticosteroid therapy treatments that are being used, like oral corticosteroid, maybe even a prednisone, if you will, to knock out inflammation, allow the healing to take place. Ultimately, you have to unload that disc. But in a lot of cases where the trauma is irreversible, and we'll show you that here in a second, you actually have to do surgical repair, where you actually cut out a portion of the herniated disc or the damaged disc, or you actually remove the disc altogether and you can put in a synthetic. So 80% of the load goes right through the disc. You're like, well, what about the other 20%? The other 20% is taken up by the tendons, ligaments, and the muscles that support the vertebral column. But the disc itself takes 80%. Some would argue it's not a great design. Maybe it should have been more like 60-40 or 50-50, because that's obviously a weak spot for us as humans. But as we move lower, which should make more sense, L4 and L5, which are more inferior as you work your way towards the sacrum and the coccyx, more weight, they're more subject to wear and tear and potential rupture. So what does it look like? Let me see if I can kill some of these lights a little bit, make this look a little bit brighter. We've got a CT scan on the left, and we have an MRI scan on the right. And what we're highlighting are the circle moon-looking structure is the actual disc. And it's on CT scan, and it's lighting up because it's primarily water, so it's going to light up pretty nicely. And this area right here that comes out away from this nice delineation is the bulging disc or the herniated disc or the slipped disc. You can see over on the far right, I got to come out a little bit further. It's hard to see from my angle. Here's our nerve root. Here's our, not our nerve root, our disc. And this portion right here is the bulge. It really should be contiguous with where I'm drawing this laser line. And this is the root, the nerve root that it's putting pressure on. So surgically, it's either a contained herniation or an extruded herniation, where you've got, it's kind of like a jelly donut. And that's a horrible analogy, but it works. So you've got a outer annulus that's kind of like a shell, and it contains an inner, more gelatinous material, which is the nucleus. And in this situation where it's contained, you just have a bulge where it's putting pressure on the nerve root. Over here, the annulus is actually torn, and this is where you've got a bulge where it's really going to need surgical repair. Here, you unload it, you lower inflammation. This will probably reabsorb on the left. On the right, you're really asking the patient likely to go through a surgical repair that's going to look something like this, where you actually will have to come in, cut out a portion of the torn, repair this annulus, or they'll take the disc out altogether, put in a synthetic, or they'll fuse the two vertebrae after they've actually removed this. You've got to do something to unload the pressure on the nerve, otherwise the pain won't go away for the patient. It's a fairly straightforward surgery. It doesn't take very long. On the slide it says, half an hour to an hour and a half. That's surgical cut down time. So that's like patient shows up at 8, maybe scalpel start is at 9 o'clock. Patient's probably in the recovery room before noon. Surgeon's on the golf course by 1. Okay. Questions over herniated, slipped, or bulging discs. Pretty common. How many have wrestled with this? You don't have to answer this if you don't want. This is a young population, and look, there's hands up. Okay, so I think maybe not the best design, right? The second one we're talking about today is called spondylolisthesis. It's a really fun word to say. Easter's coming up. You have Easter lunch or dinner with folks. Use this word so they think that you're getting a good education. Dad, I'm just worried you might have spondylolisthesis. We should get your back looked at. You're like, what the heck is that? We're going to talk about it here in a second. So this is actually an anterior displacement of the superior aspect of the vertebral column, typically over L4 or L5, where the anterior portion slides forward. Remember, we have a curvature in the lumbar. We have a lumbar lordoric angle, or is it a kyphotic angle? Which one is it? It's lordoric, right? Cervical lordorsis, thoracic kyphosis, lumbar lordorsis. So it's a lordoric angle. So from this aspect, from this view, it's actually convex or concave? Concave from this view, from the posterior view. So now, if you look at this, we've got the patient's posterior aspect to your right, and the patient's anterior aspect to your left. You can see the A and you can see the P. So this is considered an AP view, an anterior-posterior view. And in this patient, what I want you to be able to appreciate, I got to step way back to see it, okay, is right here. So you can see this is L4, this is L5. Do you see how the posterior aspect of L4 is sitting almost in the middle of L5? Let me highlight it here. From this angle, still an AP view, but they're a little bit more inferior. So they might have actually angled it kind of looking this way versus looking this way. You with me? So now it looks like the posterior aspect of L4, maybe not halfway, but definitely near halfway, where it should be lining up right here. So you get this anterior slide. That's going to put a lot of pressure on the spinal column and create a lot of pain for these patients. So what they'll do is they'll come in. They can either come in from a posterior aspect or they can come in from the belly. Because if they come in from a posterior aspect, they actually have the entire spinal cord that they have to be worried about. If they come in from an anterior aspect, right, it's all bone, the spinal cord is encased in the vertebral column, you're not worried about nicking anything and causing paralysis, but you got to move the gut and everything out of the way. Okay, so when you talk with orthopedists or neurosurgeons that do these, both will do these procedures. Some will prefer a posterior access. Others will prefer an anterior aspect. What's the difference? Well, it actually depends on their residency and how they were trained. Okay, they pretty much train one way and they stick with it for their whole career. Spondylolisthesis. Questions? Oh, this isn't here in the slides? Okay, I'll add it. Sorry about that. Then this next section is not going to be in the slides either, but I'll add it. Okay? Other questions? Oh, good question. Why can't you see the spinous processes on the imaging? They're pretty masked out by all the other soft tissue. So this is an MRI, and so you can see this really white highlighted band. It hides everything that's either in front of it or behind it because it lights up, because it's got such high visibility of contrast with respect to image contrast. And this is a portion of the nerves that are coming down the spinal column. Okay, great question. Other questions? Okay. The last little segment, and then we'll shift gears after this to the review portion. Well, we're just going to kind of do Q&A today. How's that sound? But this way, we're going to get through all of central nervous system. I think with the syllabus, these slides were actually not in your deck for this week because I moved the exam to Wednesday. That's what happened. So if you look for Wednesday's material, we would have covered this lecture after the exam instead of before the exam. Okay? So this slice here is looking at it like this, a horizontal slice through the spinal cord. You've got the anterior aspect towards the front. You have the posterior aspect towards the back. Anterior we also refer to as ventral. And posterior we refer to as dorsal. Let me see if I can get rid of that. So there's the ventral terminology, anterior, and then there's the posterior terminology for dorsal. So the reason this is important is this is a slice, just a single view through the spinal cord, and it's going to be a relay center. So there's information that's going to come, sensory information from the periphery. It's going to come into the spinal cord. It's going to be sent up to the brain. The brain is going to interpret it, like we've been learning about. It's going to send motor information back down through the spinal cord and out from the ventral aspect or the anterior aspect to effector organs. Those effector organs would be things like muscles, glands, quadriceps, hamstrings, making you run. Well, I'll hint at where we're going with this. There's also reflexes, where you take in sensory information towards the posterior aspect or the dorsal aspect. It goes up through ascending tracts. And you can kind of see color coordinated here on the left side, the patient's right, your left, patient's right. This area, this area, this area, this area, and this area are designated ascending tracts that go up. The same happens on the patient's left side. It's just not highlighted because they're highlighting other regions that are actually descending tracts that come down from the brain. So you can see on one slice both ascending as well as descending paths. But students, unfortunately, misinterpret this and say, it looks like on the patient's right side are all the ascending tracts. Right here and right here are the same areas, but on the patient's left, that are also ascending tracts. Does that make sense? It's just we're showing only these areas that are towards the anterior aspect, which also exist over here. Make sense? As descending tracts. So the way to interpret this is, generally speaking, most of the sensory information will come in the spinal column towards the back, the dorsal aspect, and go up to the brain. It goes to the postcentral gyrus. It's processed. Information is sent to the precentral gyrus, which is the motor area, and it comes down descending tracts that are mostly organized towards the front of the spinal cord or towards the anterior aspect, and then it goes out this ventral route as a motor neuron. You follow? So the general organization is, like in the brain, sensory information comes in the dorsal aspect, just like in the brain, the postcentral gyrus, which is part of the parietal lobe, is where our sensory cortex is. The precentral gyrus in the central nervous system, which is in front of the central sulcus, is designated to motor, just like in the spinal cord. The front, or the anterior aspect, is designated mostly towards motor. So sensory information comes in the back, goes up to the brain, the back part of the brain, goes to the front part of the brain, down the front part of the spinal cord, and out. Except if it's a reflex. If it's a reflex, there's an inner neuron that'll take it from here all the way to here right away, bypassing it going all the way to the brain. Well, not bypassing, sending an additional signal. It's like a group text, right? You're sending a message right away for a reflex. You send a message all the way up to the brain so it knows what's going on, but you typically move your reflex before you even realize that it's happened, because this distance is very short, and this distance, albeit through myelinated neurons, is much, much longer to get all the way to the brain. Okay? That's really the take-home message from this slide. I keep hitting this thing. I got to move it. Questions over that? So if we look at the way this structure is organized, we've got our dorsal horn. We call them horns because you can kind of appreciate how this structure is small and then it gets bigger, this like butterfly-looking print. So this is called a dorsal root horn. This is a ventral root horn. Okay? If we look at organization of lateral horns, we have lateral horns, but they're only T2 through L1. So they're not shown in this particular cartoon drawing. Things that will come out the lateral aspect. So when we look at a slice prep schematically of the spinal cord, we typically only show a structure that looks like this. We simplify it to a dorsal and a ventral root, where you've got sensory information coming in the back and we've got motor information coming out the front. But I did want you to understand, between T2 and L1 of the cord, there are lateral projections that take place. Some of the same principles fit. So if it's a white matter, it's going to be myelinated, and we refer to them as tracts, just like in the central nervous system. This is part of the central nervous system. Looking at nerves and the packaging. And this is a really important slide as we move forward into muscle physiology. Right? In lab, you're looking at all sorts of different muscles, and you're looking from a gross perspective. That means from a macroscopic view. And so I want to do a little microanatomy here, because we're going to see a similar motif when we look at a muscle fascicle as when we look at how nerves are packaged. So remember, I said to you a couple of weeks back, in biology, if you see repeated motifs, like you see a strategy being reused again and again, it's probably a good strategy to use, right? And that's why it's being used throughout the entire body. But we're way down here. If you look at, we've got either a yellow, bare, unmyelinated nerve, or you've got this sort of yellow with fat around it, so it's got a little bit of white and a little bit of like greenish blue. and this one is referred to as a myelinated nerve. And the colors aren't really important. What's important is you see that there are both unmyelinated and myelinated neurons adjacent to each other. And you're going to wrap your unmyelinated or your myelinated nerve, you're going to put a bundle of them together and wrap a covering around them called a perineurium. In a way, I feel like this looks like a complicated wire system, right? For any of you that done any kind of, audiovisual wiring, or you've seen a house being built, maybe you work on your car stereo. It's really interesting to me that this is actually, you've got different types of wire, different types of neurons, all bundled together, wrapped with a perineurium, like a casing. Now, we've got a few of these bundles, and we put them together, and then we put a sheath around it called a epineurium. One of these is referred to as a fascicle, and now, because we've gotten so big, like in bone, we actually need to have a blood supply within the nerve itself. So, once it leaves the spinal cord, it's in the peripheral nervous system. And these fascicles, if there's damage in the PNS, remember we talked about peripheral nerves have the ability to what? Regenerate. True or false? Central nervous system nerves have the ability to regenerate. False. The central dogma is that's pretty much false. There might be some unique examples in the literature of patients, one-offs, case studies. But for the exam on Wednesday, remember, we're saying the central dogma is that peripheral nerves do not regenerate and central nerves do. But this should remind you of how we organize bone. It will remind you when we see muscle physiology of how muscle fascicles are organized. Here's the scanning electron micrograph of what we're looking at here in cartoon drawing, and you can appreciate the artery-vein pair. And why do we have to have a vascular supply? Anybody want to answer that question? Why do you actually start having to put blood vessels within this fascicle? Any ideas? It's too big, it's too thick to allow for oxygenation by diffusion. And so the tissue would actually necrose unless you put a blood supply in it. That's exactly correct. Same thing with bone. It's going to be the same thing with muscle. All right. Want to look at a couple of specifics on spinal tracts. Some of this can actually go pretty quick. I think the key points that I'm going to highlight that I really want you to understand for the exam, focus in on the lecture. Focus in on what I'm saying and what I point out for Wednesday, okay? But I do want to cover the material so that you've been exposed to it, whether or not there's lots of questions on this or not. There won't be lots of questions on this particular section on Wednesday, okay? And I'm saying that because I wrote myself a note here for our review session of areas to really kind of guide you in. So don't freak out. I know you didn't have the slides yet. The stuff that I really care about is like herniated discs and spondylolisthesis and finishing out cervical, thoracic, and lumbar spine. Now we're getting into some other areas, and we're going to talk more about reflexes and how things are organized. They're not as critical for Wednesday. But for sake of completeness, I want to show you spinal tracts, okay? This is spinal tracts coming in. So this is a sensory ascending tract. For those of you paying attention to the color, this is actually the red tract that was found on that spinal slice. For those of you who don't care about the color, notice that it's coming in the posterior or dorsal aspect. So we've got receptors for body movement, limb positions. Information comes in the posterior aspect. It goes up to the medulla oblongata. It actually crossovers to the other side. That's a word that we refer to as decussation. It crosses over. it crosses over to what we call the contralateral side. If it stays on the same side, like when it comes from a lower portion of the spinal cord going up to the medulla oblongata, it's on the ipsilateral side. Ipsi means same side, contra means opposite side, decussate means it crosses over. So as it's working its way up, this is where it crosses over in the medulla oblongata of the brainstem and then continues up through the midbrain to the somatosensory cortex, which is the postcentral gyrus. We've talked about all that. Now, the descending tracts are on the next slide, and they're conveniently colored in green if you're paying attention to the colors. Now, we're sending signals down from both hemispheres to move. You got information just from one side, but you're going to send it down both through the midbrain. At the level of the medulla is where it also decussates, and information from the patient's right side crosses over to the patient's left, and the patient's left side information as it descends crosses over to the patient's right at the level of the medulla oblongata. As it makes its way down, it stays on the ipsilateral side or the same side, and it comes out the anterior or the ventral horn for motor function. Okay? Just saying the same information as we did before. Sensory information up the back side, processing in the brain from back to front, down the spinal cord. At the level of the medulla in the brainstem, information crosses, and then it exits out motor function out the anterior aspect. Pretty straightforward. So, again, just another slice that we saw before, naming these. You won't be asked for naming on the exam. Again, I just want you to appreciate the mapping of the real estate of the different areas and how they came about. These were spinal cord slices in research that studies were being done to figure out where these neurons go. And some of this information is important because we need to map the entire body. So when you have nerve damage, when you have an injury, you understand that these different plexi control different areas of the head, arms, and trunk. At the level of the vertebrae, the vertebral column, it's really more neck, arms, trunk, and maybe legs in some cases. There's a question.
So it goes CNS, which is afferent, CNS to CNS.
Uh-huh.
That's how many parts of the interneuron, right? And then it goes back from the CNS down to the PNS, which is the interneuron.
I don't know why you're using interneuron yet. Probably just because I talked about the reflex.
Well, because it's like sense, and then it gets sense with the
Oh, interneurons in the brain. Yes, sorry, yeah. So it comes up, it goes from postcentral gyrus to precentral gyrus, and those are local connections, and then it comes back down. That's correct. So it's going from the peripheral nervous system into the central nervous system through the dorsal aspect of its sensory. Then it comes up. It's processed in the brain, right? There might be certain sounds, or there might be certain feelings, or there might be certain smells, or there might be certain motions, like a rocking motion, or you feel a breeze on your leg, or you feel like grass brushing your leg. And you're like, is it summertime? You know it's not wintertime, because you see what I mean? Like all of that cognitive processing from grass brushing the back of your leg, you process in your brain. That sensory information goes to your brain, and if you felt that now, you'd wonder if you were in Southern California or down in Phoenix. You wouldn't think you were in Flagstaff. There's no grass blowing in the wind right now. So that processing, and then you might be, I'm homesick. And now it might make you think about grandma's cookies. That's how all that happens, from just grass brushing the back of your calf. That's how we interact with our environment. And that happens all day, every day. Right now you're getting tactile information from the chair. If you're wearing shorts and you feel this, 18-year-old fabric that's gnarly, it's sinking into your memory right now. The next time you come in here and sit down, you're like, oh my goodness, I'm having all these flashback memories from sitting in this chair. How does that happen? I'm telling you exactly how that happens. That's how the outside environment information, sensory, comes up, and then we process it. Not only do we send it from the postcentral gyrus to the precentral gyrus, there may be connections that go over to the limbic system in our emotional centers and our long-term memory that give us a panic attack because you felt that fabric and now you think you're taking an exam. And it's like 2029. You came here for some concert, right? Does that make sense? How all this integrates? It's kind of fascinating when you think about it from that perspective. Okay, let's talk a little bit about these plexi, and then we'll kind of wrap things up, and we'll get to review. And I want to hear from you. Now, these plexi, a nerve plexus is just a network of intersecting nerves. That's all it is. And you've got these plexi at the cervical, brachial for the arm. Thoracic is really not considered a plexus. And so a lot, because there's not a lot of nerve supply in the thoracic cavity. And this is one of the reasons if you have patients or you had a loved one that had a myocardial infarction or a heart attack, oftentimes there's a shooting pain down the left arm because the sensory input for pain for the heart actually shares the nerves that actually feed the left arm. And that's why you get a shooting sensation down the left arm. Not the right arm, not the left leg, because it's at the level of where the nerves take off near the heart. So that angina, or chest pain, which literally translates to, in Latin, strangling feeling in the chest, is interpreted by the brain as, oh man, there's some shooting pain in my left arm. Because that's the best we can do with processing that sensory information. So all these plexi are these bundles as they're organized in our cervical, our thoracic, our brachial, which is arm, sacral, and coccygeal. Okay? That's all you really need to understand or know. All right, let's ask this question and see what your thoughts are. What analogy is the best comparison to the axon endoneurium? Architecturally is where I'm going with this question. Architecturally. Axon to dendrite, Schwann cell to axon, compact bone to periosteum, perineurium to fascicle, or epineurium to axon. Axon to endoneurium. Which is the best comparison? I kind of hinted at it when I was lecturing about it. There's no muscle comparisons here. C, compact bone to periosteum. I agree. I'm just trying to make those comparisons microarchitecturally of the way things are organized. That's it. All right, last couple of slides on reflexes. And we've already talked about reflexes, but let me pose this question. The left postcentral gyrus, left side, everybody raise your left hand. It's going to be the opposite of me, right? It's going to be this one. Your left side, touch your left postcentral gyrus. Put your finger on it. Just got information about the texture of a fabric. It's obviously not the seat because you wouldn't be, right? Oh, it could be the seat. Could be the seat. It's coming from the back of your leg and you're wearing shorts because it's warm outside. Wait for it. A couple months it will be. Along which spinal nerve did this signal enter the spinal cord on its way to the brain? A, C, or A, B, C, or D, or E? It's going over to the left side. Where did it come from? Where did it originate if it's going to the left side? Let's just say that together. It originated on the right side. Okay? So it came in the right side. Is it A or B or C or D? If it's on the right side. It's A or B, correct? Because that's the right side. It's sensory information, so is it A or is it B? A. Pretty good. Good. I love it. All right, here's how reflexes work. This is pretty straightforward. So you've got a muscle that is stretched. And inside of our muscles are these structures known as muscle spindles. And these muscle spindles provide us information about how the muscle is positioned, about the length. And so they're a perfect example of a reflex. And a reflex actually has 4 stereotypical parts to it. You ready? Number 1 is it's quick. Number 2 is involuntary. You don't have to think about it. Number 3 is it needs stimulation. It needs sort of some sensory input to respond. And number 4 is it's the same every time. Exactly the same every time. Those are the 4 parts of a reflex. So if we look at this muscle spindle as an example, and we'll get into it kind of in the weeds on the next slide, you've got the primary afferent neuron. Afferent means it's going to the central nervous system. Afferent. It's sensory information going to the central nervous system. It comes in the posterior aspect. One signal goes up to the brain, like we talked about. Another signal innervates onto an inner neuron that sends it right back out the same side and right back to this structure that it came from. And that is a motor efferent with an E. And so with a muscle spindle, let's go to this slide, it's dissected away, it's wrapped around the muscle fibers themselves, and we're going to get into that in the next unit. And it's actually giving information about the length so that you don't over-contract your muscle, so you don't tear things. It's a protective mechanism. And that's the extent of what I want you to understand, especially for Wednesday, okay? It's a protective mechanism so you don't over-contract your muscles and tear something. Now, the last one that we're going to look at is another protective mechanism, which is called a flexor withdrawal reflex. Now, in a flexor withdrawal reflex, this one actually the example, you step on a piece of glass and you've got this response of bringing your leg up. This motor efferent information comes in the dorsal aspect. It wraps around, sends it right back to the same muscle group, causing flexor contractions, so you raise your leg. At the same time, you can see that you've got decussation happening, a more complicated reflex, crossing over motor efferent over to the contralateral side to extend your leg. If you're going to bring this leg up, you actually want to extend this leg to make you stand firm. Does that make sense? So this reflex, you don't do this and then fall down. You do this and you lengthen this leg and you don't even think about this response. Because this pain response with a flexor withdrawal reflex tells you withdraw this one and extend this one at the same time without you even thinking about it. Because all of this is happening at the local spinal cord right about here. And then after you do this, you're like, ow, that hurt because it finally made it to the brain. Did you get me? Same thing happens with a hot stove, right? Same thing happens when you cut your finger, you touch something sharp. Happens with loud sounds. Now that actually happens at the level of the temporal lobe, so it's not as interesting because this is all the way. I mean, this could be a sea urchin, or you're shuffling a stingray. You were somewhere over spring break. Flexor withdrawal, another protective mechanism. I lied. I had one last one. This is another final. I promise. This is the final Golgi tendon reflex. These are all really important as we move into muscle physiology. These reflexes and how they work. So this is a portion of a muscle. Let's call this one a portion of the quadriceps that's inserting into basically a portion of the femur. This is the femoral head. This is the shaft of the femur. And there's all these tuberosities and attachment sites where your muscle fibers terminate into a tendon and they insert into the bone. And at this location, you have all of these nerve fibers that are sensory input information about the tension that's happening here, the tension. So Golgi tendon organ responds to tension of the muscle. And the reason it responds to tension of the muscle is another protective mechanism so that you don't snap your muscle. So kind of the last schematic, and then we're done. Last schematic. Bless you. So here you've got our quadriceps. Here's your Golgi tendon organ. You've got one down here. You've got one up top in the origin and the insertion sites, where it begins and where it actually inserts. And you can see that you've got information, afferent information that comes in. It's going in the dorsal aspect. It's innervating on two different neurons. One neuron sends this information right back to the same muscle. The other neuron sends the information to the opposite antagonist, the hamstrings. And this is applying the brakes as these flex. Bless you. Does that make sense? Because they're opposite muscle groups. So you're getting information about tension so that you don't overuse that muscle group and cause a tear. These are all ways that we interact with our environment. If we were just straight robots and we didn't have this information, we would be far more prone to injury. Okay. Now, there's also a lot of areas of study in elite athletes where they have the capability of overriding some of these sensory inputs. And as they override these sensory inputs, they actually can go a little bit further, a little bit harder, a little bit longer. Right? And training can do that. Fight through the pain. It's really complicated because you got to get over the reflex. We are wired to protect ourselves. Right? That's our survival instinct. And so a lot of the training that many of your coaches do, without them realizing it, is trying to override some of your natural wiring so that you can go the extra distance. You can shave the extra second or a portion of a second. Is it pushing the envelope? Is it a little dangerous? Well, yeah, that's why elite athletes get injured, right? But it's also why they break records. They set PRs all the time. Okay? Questions about that? I wanted to make the link before we move after the next exam into the peripheral nervous system and into muscle physiology, because we're trying to connect the central nervous system and we're going to move it over into the peripheral nervous system and muscle groups and muscle physiology. You're studying a lot of muscles in lab. And then I just want you all to understand, the body doesn't care what unit we label it. It doesn't care that we say this is the central nervous system. The nerve, when a signal leaves the spinal cord, it doesn't have this knowledge of, now I'm in the periphery. You understand what I'm saying? We actually compartmentalize it because it's easier to write exams. It's easier to study and write textbooks and put a chapter number on it. But our bodies are very integrated. There really are no borders. There's no beginning and end. Everything is connected. We just arbitrarily draw these lines, and it's not easy to do because some could argue, why did you make that line? Well, it's somewhat arbitrary. So I'm trying to bridge that gap so it's not so compartmentalized so you guys can understand, hey, if you have a patient that presents in physical therapy with a limp and they're telling you that their ankle hurts and you're examining their knee, their gait, their hip, you're examining the other side. They said, no, it's my right ankle. Why are you looking at my left side? Well, there could be things that are happening on the left that are influencing the right. And the patient's going, well, that's dumb. Right? But you all will be the experts. You say, well, let me explain to you why. Right? The body isn't compartmentalized. That ankle injury could be coming from somewhere else. right? And as you move on in your studies, you'll start to see how integrated the body is, which makes it so interesting and so elegant. It also means that you'll have a job, because not everybody can do it, right? What's that saying? If it was easy, everybody would do it. So is it a hard class? Yeah. Do I want it to be hard? Yeah, kind of. Because if it was easy, everybody would do it, and you wouldn't be that special when you get to where you want to be. Okay, but I'm optimistic on Wednesday, as we transition into review session. You all will be, I think, better prepared, both mentally as well as emotionally. So that's all the new material I have for you. If you need to take off, I get it. I'll keep the feed rolling. Some of you told me that you had other things that you had to get to commitment-wise, so I appreciate the heads up. If you all want to stick around in the remaining time, we'll just do like a Q&A. Okay? And there's not going to be a great way to do this. It's going to be a little haphazard. I'll try my best to manage the crowd a little bit. I might ask some TAs or SIs to come up here and grab some mics and just circulate the room. Okay? If you're going to leave, could you try to do it sort of quietly? If you're going to stay, could you please remain quiet so people can listen and ask their questions? Who would like to be first with a question? Up front. And then here.
So you said for review classes, they're quick, involuntary, need stimulus, and they're the same every time. What do you mean by the same every time?
Oh, that's a great question. So with reflexes being the same every time, we would call that stereotypical, meaning, you go to the doctor, they strike your knee, and you extend it pretty much the same distance, the same force. It's a pattern. It's the same every time. It's the same motion. You don't do it halfway, right? You don't kick him in the face, right? It's kind of the same motion every single time. That's a reflex. And the Golgi tendon, the muscle spindle, the flexor withdrawal, again, same every time. The questions don't just have to be about the lecture today. The questions can be about anything over Unit 3. Here, let's pass that so people can hear. Jenna or Neve or Sasha, one of you here? I can't see you all. Or available? Oh, there you are. Go ahead. Can you circulate in the back and get some questions answered? Yep. Yeah. Oh, ascending and descending tracks on both sides of the spinal cord. Yeah, they are right and left specific. Yeah, so the right side is taking stuff up from the right side of the body. The left side is taking stuff up from the left side of the body. And vice versa on motor as it comes down. Questions on anything over the study guide up front here, Jenna? Study guide or... format of the exam, it should be old hat. It's going to be pretty much the same. You'll have like five pictures up here. Okay, back here and then right here.
I was just wondering what lectures you would say that we should rewatch to cover the test?
What lectures should we rewatch to cover our test? So the exam's going to be over all of nerve physiology. So chapter 7, nerve physiology. We had 3 lectures on that, and then it's going to be over CNS, brain, and spinal cord. We had, call it, 2.5 lectures on that. And then today's lecture probably will go live. It takes a little while to upload, so I bet in the morning it's there. Like I'll put it up tonight, I'll upload it tonight, and then YouTube has to check it. They check content, they make sure it's appropriate. I've never really had any problems, but usually by the morning it'll be up. Okay? So those are the sort of 3 plus 2.5, so 5.5 lectures that I would focus on for Unit 3. Let's talk, since you asked, great question, since you asked. What about the review material? Great question. What did I say? I think it'd be valuable to, oh, that's not review. Never mind. You can expect like 5 to 10% of review material. Max. That would be maximum. Okay. We did a review session on action potential, and then there's a question here. And if you didn't attend that, I think we ran it for about an hour. We covered the action potential for about 30 minutes. Just Q&A on the action potential. That would be a really good review session to watch if you didn't attend office hours or you haven't watched it yet. Where do you find it? That might be your next question. If you go into content, let me pull it up. I'm not ignoring you. You're next. Sorry. If you go into content on BB Learn, you'll find it under recorded office hours. Let me see if this will behave. Oh, come on now. Oh, jeepers. I love how they protect ourselves against ourselves. It's wonderful. I mean, it could be dangerous, I guess. Come on. I got to do this. See, it's not just you. I got to do it too. Go ahead and ask your question why I'm loading this up so we can maximize time. We still got like 18 minutes for lecture. We have the room, so let's use it.
I just have a question about something that's really great. What was an example of like a motor ascending signal? Because a lot of examples we have are like sensory. What would be an example of like a motor?
That's a great question. So the question is, what's another example from today on an ascending piece of input? Most of it's going to be sensory, almost exclusively a sensory input. But remember, sensory input could be body position. It could be pressure, temperature. Pressure, like if you close your eyes, everyone close your eyes. Close your eyes. Go ahead and tell me, can you tell that you're sitting in a chair? Yeah. Can you tell that your feet are on the ground? Can you tell that you're wearing shoes or flip-flops or sandals? Can you tell that both of them are on? Do you know that you have pants on today? Are you covered? Are you naked or are you covered? Your eyes are closed. Okay. Now put your hand out. Put your left hand out. Okay. Put it out in front of you. Do you know it's in front of you? Now be careful, don't hit your neighbor. Put it to the left of you. All of that is sensory information coming into you. So we think of sensory as like the fabric of the chair. That's all proprioception. That's all your limb in space. Like you can close your eyes and you can walk across the room, right? Because you know what shoes you have on. You can probably tell what shoes you have on. If they're comfortable, if they're not comfortable. Oh, I wore that pair today. I can tell that I wore that pair today. I can tell my shoe's untied. That's all sensory information. So I can't think of an example outside of sensory information right now, but I'm sure there's something else. Now, the other thing is there's sensory information that's coming that are visceral responses, like from the abdomen, from the chest, from the lungs. There's sensory information that's coming to the brain about the pH of your blood, about the oxygenation level, telling you, I need to take a deep breath right now. Right? There's sensory information about thirst, saying, I need to drink some water. And that goes to what structure in the brain? Hypothalamus. Well done. Well done. Okay, this question. Right here, recorded office hours. Now, they're limited on recordings because if I'm on with somebody and they have questions about their grade, you're not watching us navigate their grade book, okay? So there's only going to be the ones that are scrubbed This is the one that we recently had right here on action potential. It's about an hour, and I'd really encourage you to watch it if you haven't seen it yet. Okay? You don't even have to watch the whole hour. I think the action potential is like 25 minutes of the first part. Okay. Any question here? Any other questions in the back? Here, and then pass it that way for me. Where are you? Oh, there you are.
When hyperpolarization happens, and I'm looking at one of the from the PA, it says that the polarization occurs when ion channels open and allow left to enter and right to leave. Could you please?
Say it a little louder, sorry. When action potential does what now?
When hyperpolarization occurs, do ion channels open and allow cations or anions to enter and one to leave?
Okay. When hyperpolarization occurs, just so everybody's following, we're talking about number 6. Okay? And I think what I heard you say is, this sounds like a TA practice exam or quiz or something like that. Again, they're writing content. They're in lecture, right? We meet every Tuesday. So they've taken this class. They've all gotten, I'm sure this is a violation, but they all got A's in the class, okay? So they know what they're doing. They're not writing the exam. So some of you ask, like, why do I feel like it's the practice exam and I know all the stuff on the study guide, and then I get to your exams and I don't do very well, right? And I'm going to get to your question here in a second. Because we're writing things in a different perspective, and my goal is not to see if you can memorize something. It's to actually test if you actually really understand it. I'm testing you for comprehension, not memorization. Because if it's just memorization, it's going to go from short-term and stay in short-term memory, and you're going to lose it. If it's for comprehension and you use it and understand and it makes sense, it's more likely it'll move over into long-term memory and stay. And if you're in this class because you want a career in this field, whether that is science or medicine, you're going to need that. And so that's my mission. That's my job. My job isn't to hand out good grades. My job is to help educate you. And so I'm trying to share that with you. So if you are frustrated, and you can be frustrated at me, and I get that, but understand I'm actually trying to do this for your benefit. Okay? And if you stick with me and you lean into it, I think you'll get there. I really have confidence that you'll get there, and we'll be on the same page. Now, at hyperpolarization, the question is I'm paraphrasing, cations or anions responsible? What's happening at hyperpolarization? Okay? Anybody want to take that question and see if they can answer it? What's responsible for 6 hyperpolarization? In that office hour that I pointed to, we go over it, but we're going to answer it right now. But does anybody want to take a stab at it? Okay, Kai? Pause. Can you pass that to him? Next up, you're coming up here, and I'm going to have you teach. Just kidding. Okay, what's responsible for number 6? Start over, sorry.
Potassium slow channels, they also close for than the sodium channels did. So there is more potassium that leaves the cell than sodium that enters the cell. So the resting membrane potential goes lower than the original resting membrane potential until the potassium channel closes.
And the sodium potassium ATPase reestablishes resting membrane potential. Okay? So, for example, Kai's totally right. The sodium channels, which are cations, so to answer your question, it's all cations. Anions have nothing to do with it. So don't get confused by that. Because it's totally like me to say chloride ion is responsible for doing something here. You guys know what I'm talking about, right? But you're not going to get confused about Keller putting chloride ion as being any culprit here, because you understand it. We're moving positive charge cations. So at number 1, these are local potentials that are small. They can summate either temporally or spatially to hit minus 55. Once they hit minus 55, which is less negative than minus 70, this is a depolarization. Polarization means it makes it more different, more polarized. We're making it less polarized. We're depolarizing it to minus 55 at point 2. At point 2, the fast sodium channels, like Kai talked about, those fast voltage-gated sodium channels open. And their trigger is a minus 55 voltage. They open, and that spike of 3 is really fast. And then they close at 4, and that's why it makes a U-turn right here, really fast at 4. Because the fast sodium gates shut, they open fast and they close fast. Now, Kai is totally right. The slow voltage-gated potassium channels also start to open at 3 on the upswing. They stay, and they're fully open at 5. And they take longer to close. That's why they're called slow-gated. And so that's why at 6, because they're closing slowly, you get this overshoot. You move too many potassium ions, which direction? Out of the cell. Because they're positively charged, you take them out of the cell, you leave a negative behind, inside the cell. That makes it more negative, down to like minus 80-ish. And then, once they close, and then the sodium-potassium ATPase takes what? 3 sodiums which way? Out of the cell, and 2 potassiums which way? Into the cell to establish the gradient. That eventually brings things back to homeostatic norm, which is minus 70, and that's at 7, resting membrane potential. So if that was too fast, if that was confusing, please watch that recorded lecture, because for 25 minutes, that's what we talk about. And that's the only thing we talk about for like 25 minutes. Are you all with me? Okay. Do you have another question back here? Okay, one more question, then we'll pass it here, and then we'll go here.
I watched the lectures up till now to study for the test, and I got confused as to where cerebrospinal fluid is made, because you said it was made in the subarachnoid space, but also in epidermal cells in the central nervous system. So I'm confused.
Okay, fair. So maybe I misspoke. Let me unconfuse you. Cerebrospinal fluid is made in the ventricles. It's made specifically in the choroid plexus, which is a part of the ventricle where the ependymal cells line. The choroid plexus is a network of vessels where the blood supply in the ventricles, the lateral, the third, and the fourth, where the ependymal cells can help filter the blood and generate a clear, odorless, colorless fluid that looks a lot like blood plasma without a lot of the protein. So it's not quite as yellow as plasma. It's got a slight yellow color, but it's mostly clear. And then it circulates through the ventricle system, and it moves into and circulates through the subarachnoid space, but is not manufactured in the subarachnoid space. And then it drains in what we call the arachnoid villus into the venous circulation back into the bloodstream. And that is located within the subarachnoid space, the drain. So the faucet, the start is in the ventricle, the drain is in the subarachnoid space. Okay? If I mixed that up or confused you, I apologize. Okay, back here.
So on the study guide, there is a question about the neuronal circuits. So I'm wondering, kind of like what the purpose of them is and what each one's used for.
Okay, great question, because I think you're going to want to know this. Oh, where's all my... Hang on. So we had a couple of circuits that we talked about, like converging, diverging. Is that what you're talking about? Okay. It looks like some of them got deleted off of here. Oh, wow, that's going fast. What's your question? Can you ask your question while I'm pulling this up? It's back to action potentials. Okay. What about action potentials? Okay, say that a little louder. I'm having trouble hearing. Sorry. That's correct. They're both cations. Oh. You're talking about the gradients. So you're talking about this slide right here. Yeah. So yes, that's part of the answer. Yeah, but remember that the sodium-potassium pump pumps sodium out of the cell, so it's high outside the cell, and it pumps potassium inside the cell, so it's high inside the cell. So potassium's held inside the cell kind of against its will because of the sodium-potassium pump. But it's necessary because when you open voltage-gated channels, you need sodium to come in, right, to create the depolarization, and then you need potassium to want to leave. But it's going to leave a little slower, which I think is what you're pointing out, because it's working against one of these forces, whereas sodium has both forces working for it to go into the cell. You all with me? So voltage-gated sodium channels open, it really wants to go into the cell because it's going down its diffusional gradient as well as its electrical gradient. Potassium, on the other hand, it's high inside the cell, so it wants to leave, but it's positively charged, so there's the electrical force it's kind of competing against. But that diffusional force is so strong, it will leave. It's just not going to go as fast as sodium. They run all day long. They actually are running while you're sleeping. They use actually we talked about that. They use a significant amount of energy in our body. They're always operational. Okay, I don't know why this happened, but my neurophysiology lecture, I lost it on this computer, so I had to pull this up. So you want to know about circuitry, and I think that might be our last question. I can hang a little longer until we kind of get kicked out of this room. That's fine with me. But if those of you that need to leave can, maybe try to go quietly. And let's just get to circuitry. This one. Is this it? Okay. All right. We've got a couple of different types of circuits. Oh, wait. This is just IPSP. Oh, neural circuits down here. This is important. I should talk about this. Okay. Do you have a second? Okay. So we're talking about summation. Why is summation important? Because summation has to happen for us to get to threshold. And so here you're seeing excitatory EPSPs. You're seeing excitatory EPSPs. There's 3 of them with arrows pointing to them, adding up, stacking on top of each other to get to the minus 55 so that you'll fire an action potential. You can either do that on one nerve, and you can fire the nerve once and then fire it right away a second time. And you stack it before the first signal dissipates. And that's called temporal summation. You're adding, that's summation, you're adding over time, temporally. So you're stacking one signal on top of another. Okay? Or you can actually add what's called spatially, where temporal summation is the top graph on the right. And that middle neuron has these, see these lines across it? Every line is electrical activity. So there's like, I don't know, 10 dashed lines. the circuit above it and the circuit below it have nothing. It's just that middle one. So it's temporally summating right here until it has enough of a signal to reach minus 55 to actually fire the neuron. That's an example schematically in a cartoon of temporal summation. Spatial summation down here, the bottom one, you got 3 different neurons that are firing not nearly as fast. You see how much time space is between the lines? So this hash and this hash are further apart. This hash and this hash are really close together. That means that these are not being fired as quickly. But you've got 3 of them doing all the same thing, and it's spatially from 3 different locations, spatially adding up or temporally summating. Spatially summating. Okay? So that's temporal versus spatial summation. And I talked about it in a couple different ways. Like I talked about this figure right here, right? And just to dissect this one, just real quickly, if you look at panel A, like on the far left, panel A in pink, you see an EX1 and an EX1 that are green. If you go up top, the very top neuron that's green that says EX1, that's an excitatory neuron, number 1, that we've labeled. And that's the one that's active, and it fires, and then it dissipates, and it fires again. So the second time it fires is after the first one went away, so nothing happens. I'm right here. This one fires, and then it rests, and then it fires again. So it didn't temporally summate. In B, panel B, here it fires, and before it calms down to baseline or resting membrane potential, it fires again, it reaches threshold, and that's when the sodium gates open and you get an action potential. C, you've got, same thing, you've got EX1, and then you have this black one, EX2, right here. So these are 2 different neurons. It's not going to be temporal summation, it's going to be spatial summation because it's 2 different locations in space. You with me? So now this one fires, EX1 fires, EX2 fires. It's kind of the same result as panel A. Nothing happens. But if you bring them together, EX1 and EX2, at the same time or nearly the same time, you get an action potential. Panel D is showing they can cancel each other as well. So now we introduce the complexity of an inhibitory neuron. Inhibitory neuron 1 in red at the bottom. That's responsible for inhibitory 1 and inhibitory 1 here. And if excitatory fires and inhibitory fires, it cancels itself out. This is how we regulate. Have you done an activity like I think the best, I'm not a swimmer, but the best example I can think of is like swimmers. They're on the block. They're standing over the pool. The pool is glass. They're all ready. Like a false start with a swimmer is like pretty obvious. Like the pool is all, disturbed. You know what I mean? And they're like right on the edge, right? I don't think they can even flinch. If they flinch, I think it's a false start. There's so much of this happening with that swimmer or a sprinter or a hurdler or a football player, right? On the line, defense, offense. Like you are ready to go. It'll be you on Wednesday. You'll be like, say it, Keller, I'm ready. Right? You're ready to go and you're just poised. That's all because of these synapses. You've got inhibition saying, don't go yet, but be ready, right? It's all that prep work. I mean, athletes are probably some of the easiest examples for me to conceptualize, but there's lots of examples in our daily life. Did that answer your question? Okay. One more? Any more? One more here?
So on the study guide, it asked what happens when it reaches the synaptic knob, and I said, calcium channels open and the ions diffuse. Is that correct?
Say that one more time. It says what?
So the question was, what happens when it reaches the synaptic knob?
Okay.
But voltage gated calcium channels open and the ions diffuse. Is that correct or no?
So the question is, on the study guide, what happens when the signal reaches the synaptic knob? So I think the best answer would be, if you're at a synaptic knob, you're going to have to go from an electrical signal to a chemical signal and then back to an electrical signal. So you need to release a neurotransmitter to cross the cleft at the end of the synaptic knob. That neurotransmitter needs to bind to its receptor on the postsynaptic membrane and cause voltage-gated sodium channels to open. And when they open, now you propagate the electrical signal on the other side of the synapse. Does that make sense? Yeah. Okay. Yeah. Oh, yeah. Yeah, the question is on the herniated disc, can we say that nerve is being pinched? Yeah, or impinged is the clinical term for it. You have an impinged nerve. Yep, and it's shooting pain. It's debilitating in a lot of patients. And it doesn't matter, like some positions are better than others, sitting, standing, but most of the time patients will say, like, I can't get any relief. And you know, physical therapists will do traction, like lumbar traction, and usually now it's in a supine position, so you lay down on a table, and then they put you in a machine that kind of straps a harness around your waist and one across your chest, and then they just slowly kind of pull you apart. Like the old days of like putting boots on and hanging upside down are kind of like, 1980s. But now the traction is a little bit more sophisticated. They're pneumatic. You can regulate how much pressure you're putting on the patient. You can actually heat with pads. You heat the back first with warming pads so that you loosen all the connective tissue before you pull. And you don't like, wah! You're just like, you're pulling very slowly with like a pneumatic pump. And then you hold. And then you might put a little bit more pressure and hold. And a lot of physical therapy for vertebral problems involve traction that looks like that now. Okay, but if it's torn, annulus, and you've got the nucleus purposa poking out, and you can't really get the jelly back in the donut type of thing, they'll come in and surgically remove that. Sometimes they might even try to open up the bony foramina that the nerve comes through, just to give it a little bit more, especially if it's inflamed. Because if it's inflamed, the hole that it used to fit in doesn't fit anymore. So the surgeons will come in, they'll use anti-inflammatories to try to reduce the inflammation of the nerve, but they'll also come in and open up with a formectomy, open up the foramen. Those, all these holes and tuberosities and things that you study in lab, like they actually have a purpose. Like any hole usually means something passes through it. And a lot of them are nerves. And so if the nerve's inflamed, you either shrink the nerve or you open up the hole or you do both. Just depends on how aggressive the surgeon is. Okay. Anything else? There must not be an invented here, so they haven't kicked us out yet. In the back, yeah. Okay, hang on. Let me pull it up. I think I know which one you're talking about. I think we're looking at unipolar, bipolar. This one? That one? Yeah, so you can either classify them by structure or function. So this classification is by structure. Okay, we're classifying neurons by their architecture. So we have multipolar up top. Here are my notes. You can see multipolar up top, which are most of the neurons in our body, where they actually have, a number of different dendrites, not just a single, version that we draw. Like the bipolar neuron is what the textbooks like to draw. This is like the model, pretty-looking neuron, right? This is all of us. We're not the models. We're just regular people, right? So this is like 99% of the neurons in your body are multipolar. Bipolar are what you kind of see in the textbook, where the cell body is right in the middle. You got an axon on one side and the dendrite on the other. And then this is the unipolar, where it's kind of on the side. And then I think I gave some examples. Bipolar is retina, olfaction. And then I gave some examples of like unipolar primary senses, like light, temperature, and sound. But the vast majority are multipolar. And then from there on out, we really kind of classify them from this class by function. We call them either sensory, afferent, or motor, efferent, or association neurons like interneurons. And even now, I've stuck with that nomenclature by function, how they work. Does that help? Okay. Yeah. Oh, pseudo-unipolar and unipolar. I didn't hear that part, sorry. So I guess the pseudo-unipolar would be where there's debate on how much of a side branch there is. You with me? So it's probably at what level, how close does this get between variations of a bipolar? If it's sitting right here, it's bipolar. But if this is really, really short and just sitting right here and this is a stub, then we're kind of on that cusp of, pseudo-unipolar versus truly unipolar. It's literally just a classification system. I can promise you there won't be a question on the exam on pseudo-unipolar versus unipolar, if it helps. Okay? I won't split hairs like that. That's a good question. The question is, why do the central nervous system neurons not regenerate? And I mean, the short answer is we don't really know. And we've been trying to research it, the collective we, as a field, have been trying to figure out why. Because we get regeneration in the PNS, peripheral nervous system. We've got a lot of the same parts available to us. Like we have myelin in the CNS, we have myelin in the peripheral nerves. And the myelin in the peripheral nerves is actually what forms that regeneration tube. But in the CNS, it just doesn't regenerate to the same level. One of the theories in the CNS is the real estate in the CNS is defined. What I mean by that is you're born with the neurons that you're going to have in your central nervous system. And slowly, even at this age, while you're young and spry and vibrant, you're losing neurons on a daily basis. Mostly you're losing the central nervous system neurons that you're not using. And the ones that don't get used die through a process known as apoptosis. They just sort of are signaled for cell death, and their parts are recycled. Your body's saying, hey, if you're not using it, I'm going to use the machinery and the energy somewhere else. If you want to go back and relearn something or learn something new, the question is, well, what happens if you need it later? Well, there's a lot of neuroplasticity, and so you can reroute through different pathways in the central nervous system. And that's kind of how a stroke patient recovers. So if a stroke patient has a massive stroke to the cerebral hemisphere, motor function is severely compromised, and they have a kick-butt physical therapist and neurophysiology team, and let's say they get back into like regular life, they have no awkward motor control of the hand, they don't have a droopy face, they don't have a drop foot when they walk, everything is working well. They have rewired their central nervous system to use neuronal pathways. It's kind of like your maps saying, at your next convenient opportunity, turn around, you dummy, right? Because you missed your turn. And it's like, oh, take a left here, take a left here. It's your next opportunity, take a right, because now it's got to go all the way around here for a U-turn because they're all one-way streets. Your brain will do that to some extent. Now, the ability to rewire or remap, if you will, as we age is severely compromised. It's much easier to do it when you're younger. So you see like central nervous system patients have traumatic brain injury from motorcycle wreck or bicycle wreck or something like that. If they're young, the chances of them recovering with PT is much better than a stroke patient who's in their 80s. But it does happen in the elderly too. So going back to your original question, because that's the way it is, I kind of had to explain that to get to my point, is your brain kind of shrinks, it atrophies as you age. The size of your brain actually atrophies. You have less convolutions, you have less mass, you're taking up less space. And you've learned more information. So if the opposite was true, if you added neurons to your brain and your mass increased as you aged, we would actually have fontanelles that we'd have these monster alien heads. So there's a real estate issue. I mean, you have a confined amount of space where you become more efficient as you age with the neuronal circuitry that you have versus actually adding more neuronal circuitry as you get older. You with me? So that's more of a spatial architectural argument that's in the literature today. It's controversial, but it does make some sense. In the periphery, you don't have anything. So what am I saying about confined real estate? You have a cranial cap that fuses by the age of 3. Everything's closed up, right? All the fontanelles, the soft spots are gone. And then the spinal column, like your vertebral column, you get a bigger spinal cord. It's not like your vertebrae get larger, right? And you're like, well, Keller, I thought you said the bones would remodel based upon the stresses placed on them. That is true, but that's internal architecture, not necessarily size. Okay. Does that help? Apparently the biology evolutionary didn't see a need for it. And so we've conserved the ability to do that in the periphery, and we've let it go in the central nervous system. Yeah. This is feeling a little bit more like a TED Talk now than a lecture. Okay. Which is fine. It's totally fine. I've got time. So if you guys have more questions, I'll stay as long as they'll let us. Or as long as you guys have energy. Yep.
So if ionotropic nervous is faster than metabotropic?
Metabotropic, yep.
What is the advantage to doing metabotropic?
Yeah. It's a control benefit. So with metabotropic, okay, so the question is, you got ionotropic and metabotropic. See, these questions tell me, and this level of commitment tells me that these are going to be some good grades right here that are staying, right? Or those that watch. Because if you're asking these level of questions, you're going into the detail, and you won't misunderstand the question on the exam. And if you have a question on the exam, please raise your hand. And if you want to be, a little, persnickety, and you want me to come and answer the question, I'm fine with that. Just be patient, because there's a lot of you. We'll get to you. But most of you got plenty of time. When we get to the end of the exam, there's like 2 people in here at the hour 15 mark. So you got lots of time. I'm optimistic on Wednesday. I'll be telling you all like, hey, we kind of have to get out of here in about 5 minutes. So let's wrap it up, right? So back to the question. Ionotropic versus metabotropic. I think the video cuts off. Like, I got to stand right here. Ionotropic versus metabotropic. If ionotropic is much faster, which is true, and metabotropic takes longer, and we're talking about neurotransmitters and the classifications of neurotransmitters. When we get to we're at the end, we're at the synaptic knob, presynapse. We're going to the synapse, and then we're going to go to the postsynapse on the other end. And this unit is neuron connected to neuron. Next unit, it'll be neuron connected to muscle. So it's going to be the same stuff, just now it's a muscle on the other side. And it gets really exciting over there, okay? So why use ionotropic if you need it to go fast? Ionotropic goes quickly. Metabotropic, because it's the neurotransmitter like acetylcholine that goes the other side and stimulates ions to move. like voltage-gated sodium channels to open, and now you get your action potential again. So if that works so quickly, why even bother with metabotropic, which is essentially the question. Am I right? Metabotropic sends a signal to the other side. It sends a second messenger system that gets a piece of information to the nucleus of the cell on the other side. And now you have control. You can say, hey, this signal just came through. What do you want to do with it? So it's a level of control that we will use on certain synapses. So it's not always a knee-jerk reaction, or, sometimes it's not always, my mom always said, just because you can doesn't mean you should, right? And so our body says kind of the same thing. Just because you can act there, maybe we should kind of think through this a little bit, and that's what the nucleus does, because it's the decision-making part of the cell. It gives the cell information about what's happening. If that signal is happening on a regular basis, then the sensitivity of that pathway may be heightened. Let's say it's a training operation. You're trying to train a new technique. Well, why do you get better at certain activities when you practice? Because those neuronal pathways become more sensitive to that activity, right? And that circuitry, if it's well used, can almost anticipate what's happening. You can only do that if you have information to say by the cell, hey, we seem to be doing this a lot. Maybe we should recruit some other pathways to help us with this activity. And that's how you reinforce that ability to throw a fastball or a curveball or jump at the right time from the swim platform, okay? Or make the turn in the water, right? Or swing a baseball bat, or play the stanza that you can't get through, and you practice and you practice and you practice, and now all of a sudden it clicks. Okay.
Swarm cells are, for lack of better terms, the cells that are inside the myelin sheath, even though they're technically on top of it. Then what kind of cells are actually inside or on top of the myelin sheath in the central nervous system? The swarm cells aren't in the central nervous system.
No. Oligodendrocytes. So,
Make the myelin sheath, but are they on top of it?
yeah. Yeah. So if you go back to, I showed a video, which was a pretty good video, where imagine, I kind of think of it like making a cinnamon roll. Or think of it like a snail. Okay, like a snail is gonna move, and it's gonna leave like a snail slime, and you physically can roll that, and you can roll that slime, and eventually you'll get to the snail, right? Or a cinnamon roll where you have an end, and let's say you put like a raisin or a piece of chocolate on one end, and now you're going to roll the cinnamon roll until you get to that round structure, and it's going to roll in and fold onto it. That's kind of how the myelin sheath wraps around the axon. So the tail kind of the cell, it's like a long cell. It's a cell with this long tail, and it's just a long piece of membrane. It's still part of the cell. Because it's a membrane, it's a fat protein wrapping. That's why it's considered a lipid protein layer, because the phospholipid bilayer of the cell has transmembrane proteins inserted into it. So if you roll that, you don't just get fat. A lot of people misunderstand. A lot of instructors teach it wrong. It's not just a fat membrane. It's actually a fat protein membrane. Make sense? So it's just like the end of the cell that it grows kind of this long membrane. It says, all right, well, you use this and insulate. Does that help? The myelinated and unmyelinated tracts are very well mapped, and they're pretty much the same in all of us.
And there's like both for, say, one versus one versus the other.
Yeah. So, generally speaking, for the purposes of this class, just to kind of keep it at a 200 level, if you have a long distance to go and you need to get there quickly, you myelinate it. If it's a short distance, because Kai kind of asked that, or somebody, you asked the question, kind of like your question of like, well, if you can myelinate it, why don't you just myelinate the whole thing, right? Well, because sometimes you don't need to. So if you're going short distances, if you're going post-central gyrus to pre-central gyrus, they're literally right there. There's no need to myelinate that. Even to the limbic system for memory, you don't, or emotion, you don't need to myelinate that necessarily.
One jump from one node to the other over the myelin, that's one action potential, correct?
So the action potential is the recording of electricity if you were to insert a probe just beneath the membrane of the axon. So the axolemma. So you're just inside, just beneath the surface. And that's where your electrode is. And you're measuring what that electrical signal is. No matter where you measure along that membrane that's not covered by myelin, you will see that action potential signature. Where the myelin sits, and this is kind of interesting. You imagine all this activity is happening inside the cell, inside the axon. The myelin is wrapping the outside. So a lot of these videos show the action potential hopping outside. It's really more accurate to say it actually hops underneath the myelin inside the cell to the next spot where there's actually a voltage-gated sodium channel. And then that opens. Because you're measuring activity inside the cell, not outside the cell. Does that make sense? So it's easier to draw it on a video of it going boop, 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 boop. But it really is happening underneath the myelin inside the cell. And it's just cruising inside the cell until it looks up and there's no fat protein layer. and then it'll initiate a voltage-gated sodium channel to open, and sodium will come in.
At the beginning, we started with more potassium inside the cell and less outside, and then vice versa with the sodium. At the end, there's more sodium inside the cell,
Yeah.
and so that, the end of that electrical signal, like you just said, is what shocks the next one. It doesn't go back to being more potassium inside and less sodium.
It does. What's responsible for resetting it when everything's over? When everything's over at that location on that axon, what helps to reset those ions to where they need to be? Do you know? Anybody can help her?
Because I know there's the hyperpolarization,
Yeah.
and then the resting membrane potential goes eventually back.
And what brings it back to resting membrane? Kind of reorganize, cleans the room up, puts it back where it's supposed to be.
Okay.
What is it?
Doesn't it depend what it is regarding CNS symptoms?
Nope, it's pretty universal. Help her out. Yep.
So at the end,
Yep.
it's exactly how it started.
It takes a few milliseconds, but yeah. Yep. Those pumps are constantly going. Have you guys ever seen like a sump pump or been on like a big boat on a lake? I don't know if anybody boats a lot, but like up at Powell, they have we have big boats up there, and their big boats have like constantly are taking on water. So they have bilge pumps. Like you'll walk down the pier and you walk down the marina, and it looks like these boats are going to the bathroom. There's like this constant flow of water pouring out of them. Like streams. Kind of, I know it's a funny kind of analogy. But those are the bilge pumps just constantly pushing water out of the bilge, out of the bottom of the boat. That's kind of like your sodium potassium ATPase. It's always running. Yep. It's always running. Yep. Yeah, so that was the disconnect. Is you're thinking it shuts off when this thing's moving. They're always running. There are other channels and there are other things happening at the membrane than what we've taught in this class. I'm just boiling it down to kind of a 200 level basic so you can understand the principle. And then in your next classes, you'll bolt on more stuff. You'll learn about other channels and pumps. You're like, oh, well, Keller never told us about those, right? So we're ignoring chloride ion. We're ignoring calcium. Somebody asked about calcium. Who asked about calcium? Oh, she left. Okay, we'll talk about calcium when it comes to the muscles next unit. We'll talk about muscle physiology. Calcium we cannot ignore when it comes to muscles.
In the review session for action potential, we were going over how the potassium pumps start to open at 3, and then you said at 4 they're fully open, but today you said they were fully open at 5.
The potassium? Yeah.
Because on the, like, on your actual slides it says they're fully open at 5, but in the review.
Okay, hang on. Let me pull it back up here. So here on the repolarization size, I mean, some of it's a little splitting hairs, okay? I think, especially, so here's a great example. If you know this stuff, and I wrote a question, and you come and you see your test, and you're like, hey, well, wait a second. That's kind of like splitting hairs. Like, I want to explain to you how I understand this and how you ask the question. And if we want to have that dialogue and you convince me that, yeah, that makes sense, then you'll probably earn those points back. Does that make sense? So at 2, everyone's on the same page that fast-gated sodium channels open at 2. They're really quick. Okay? 3, they're open. They're fast. That at 3 is where the potassium gates start to open too. They got to get ready. They wake up slow, So they got to be active over on 5, and over on 5, the sodium gates are closed. They close at 4. Sodium gates close at 4. At 5, they're already closed, and the potassium gates that opened over on 3, they're still open at 4. They remain open at 5, and they don't close until 6. Does that make sense? So depending upon how I ask the question, I think you can split hairs. And so don't get frustrated or worried about that. Like, I've tried to write these questions where they're not going to be confusing. If you understand it, you should be able to explain it. And like, for example, if I ask a question about like what's happening with chloride ion, some of you should chuckle and go, there will be students that miss that because they didn't get into the weeds, and chloride ion has nothing to do with this graph. So you can eliminate that answer on the exam, right? And some students are going to be like, it was so confusing. I studied only sodium and potassium, and then you threw out chloride ion. Do you see why I do that? Like, if you know the material, you won't get distracted by that stuff. Does that help? Okay. Oh, this is a great question. Local potentials happen and summate and accumulate at what location on the nerve? It's not the dendrite. What is it? Anyone? Trigger zone, another name for it is axon hillock. Very good. Yep. So right here, like this funnel. Make sense? For the action potential. But that's where local potentials that come in, they can come in from any location on those dendrites. They come in, right, and then they summate at the trigger zone. If it hits minus 55, you send a signal down the axon. If it doesn't, it just dissipates, and then the sodium-potassium ATPase puts everything back in order. It's a lot of energy to do this, right? So do you all remember me telling you when we talked about sodium-potassium ATPases and how much energy it took, and how I told you like studying and intellectual challenging activities burns a tremendous amount of calories. Like if you study for a couple hours, you're like, I'm hungry. You will be. Like, this is so much energy, and this is happening right now. All of us are doing this together, right? And so I said, I mean, some of these things, like, I didn't see any signs. I don't know if they don't want food in here, but like, if you need like a bar or, like goo or shot blocks or liquid IV or a thing of tea or coffee, just don't make a mess, please. But if you need something on Wednesday, just to keep the distractions, keep your energy level, lower your nerves so you're not so anxious, bring it in. Earplugs, some students are going to be bringing in earplugs because they get really distracted when people are getting up and leaving. Okay? If it's like this and we have 20 minutes left and this is the number of students, look around and you just want a little bit of space. You say, can I go sit back there, Dr. Keller? Just raise your hand. Can I sit up front? I can't see the screen as well. I forgot my glasses. Just whatever we need to do to take the anxiety. Some of you are like, I have tests taking anxiety. I know this stuff. But the dialogue, this kind of stuff this is how I think you should be studying, by the way, with each other, asking each other these types of questions. Tell me about this. How does this work? Tell me about this. Not memorize this, memorize that. Like, you will never remember that come fall when you take 202, if you just memorize it. If you understand how it works, you might need to refresh yourself come fall, but it'll come back much quicker. Be like riding a bike. Somebody have, yep.
For Broca's aphasia, I think like Broca's area is the area of the brain where you can like actually talk, right?
Mm-hmm.
But in Broca's aphasia, you can't talk. You can only understand like words and like break words. So is that because the Broca's area is damaged in some way?
Yeah, so Broca's is towards the frontal cortex, so more of the motor. Wernicke's is towards, this is on the left side. Wernicke's is more towards is in the parietal lobe, which is more sensory. So with Broca's, it's a speech formation problem. Like, I want to tell you. I want to tell you something. So they're having trouble with the motor function, a lot of stuttering. And then the Wernicke's is they don't understand what they're saying. They'll say stuff they shouldn't say, inappropriate stuff. Like swear, cuss, talk like a sailor. Sorry if anybody's got sailor for parents, but that's the saying, That help? And it's all because of where it is in the brain. So that's another piece too, is that's why I show the brain maps and we talk about what it does. It's kind of boring information if you just think about it like, well, I just got to look at this weird looking map with all these numbers on it and just memorize where stuff is. But if you study it with, okay, what happens if there's an injury there, or if there's a stroke there, or it's not, you have a hemorrhage, a contusion in that region. You have a tumor that shuts off that part of the brain. Think of it that way. How's the patient going to respond? What's the clinical sequela? If you've got the pituitary gland, for example, and then I'll get your question. And, somebody's got an injury in the pituitary gland. Could one of the results be that they, just grow like crazy? They have an overactive tumor and now all of a sudden growth hormone is high? Yeah. What happens if the lesion is in the occipital lobe and it's on the left side? And could they lose right eye vision? Yeah. They'll lose a portion of their left vision because it hemidesiccates. Like the occipital lobe sends it to the other side and keeps some of it on the same side, right? Because vision is so important to us, like bilateral vision. So we take images from the right eye and we send it to both lobes. But the right eye mostly goes to the left side with a little bit of information to the right occipital lobe. But that's the way to study that stuff is, because I'll ask questions like that. I will. I'll say, hey, a patient has a lesion here or has this problem in this location. Which of the following would likely be a result? And you all will chuckle and be like, yeah, no, I get it. I get why he's asking it that way. That's what you're going to be doing anyways in the future, right? Okay, back here.
So with the Wernicke's area, that's the understanding of written and
Yeah.
So with the Wernicke's aphasia, how if they aren't able to understand that like written or spoken language, do they not like do they remember those words to repeat?
Well, because they're in other parts of the brain. Yeah. So trust me, by the age of, by seventh grade, you've heard it all. You've heard all the words, right? I only have 3 teenagers now because one of them's in their 20s, but, and they're good kids. But I remember the first time as a parent, we were watching a movie and I was like, oh no, this is horrible. And my youngest was like, Dad, I know all those words. I was like, really? She's like, yeah, I go to public school. I was like, okay, cool, play. Yeah, so it's somewhere else in the brain. It's probably in the limbic system, housed in either an emotional center or in long-term memory. Yep. It's a good question, though. Like, how can they do it? Because it's not the only part of the brain that's operating. But it is the part of the Wernicke's area is the part of the brain that's following what you're saying and comprehending it. So it's operational right now because you're all asking questions and I'm saying stuff and I'm hearing myself say it and I'm thinking, oh, no, that's not right or that's right. And I would correct myself, right? Which is fascinating. Think about that. When someone asks you a question and you're able to answer that question, even as simplistic as it is, customer service, what's your name? What's your address? What's your phone number on file? Right? Oh, it's not coming up. I need a password. What's your pet's name? Mother's maid, all this stuff, it's stored somewhere. And you're having to recall it. Wernicke's and Broca's are both firing at the same time because you're recalling information from other centers in the brain, long-term memory, and it's moving it to Wernicke's and Broca's. You're saying it, you're understanding it, and everybody's happy. If you cuss the lady out on the phone, right, something's wrong up here. Right?
Still be different, even though they have the exact same DNA,
Mm-hmm.
and one might argue the exact same brain.
Mm-hmm.
So how is that possible?
Yeah, I have no idea. It's a great one. That's research you should go into. Identical twin research. Yeah. Yeah, so it's fascinating. We'll end with this one. The identical twin research, it's the nurture versus nature discussion, right? Where, and they've done studies. These are case studies, and it's an N of 2 because it's a pair of twins, and they have the same DNA, so they're genetically identical, but they were raised in 2 parts of the country or 2 parts of the world by different families, and they come together, and it's amazing at how different their lives are, or in some cases how similar their lives are. And the same thing, they could be under the same household, raised by the same parents, have the same experiences, and they have different interests and different tendencies. One likes sports, the other one doesn't. It's a mystery. We think we've unlocked it and we figured it out, and we know very little when it comes to neurophysiology and kind of brain mechanics and how it works. That's one of the reasons that's not my area of research. I think it's overly complicated. I don't think I'm smart enough for that stuff. I stick to kind of simple things. I need to understand it to be able to be passionate about it. And so if it's hard for me to wrap my head around it, then, I kind of move on. But I don't have a great answer for you, Kai. It's a great question. Yeah, it's a, mystery wrapped up in an enigma. It's a great question, but there's a long list of questions I have that have never been answered. So, well, thanks for sticking around. I am thoroughly running out of gas, so I think we'll adjourn this longer than usual lecture review session. Did y'all see that I have more office hours this week? Because the exam's not till Wednesday at 4. So on Wednesday from 10 to noon, I'll be in my office or on Zoom.